Chapter 12, Section 4, Active Reading Review. Number 1, recall the parts of an operon. The parts of an operon include an operator, a promoter, a regulatory gene, and the gene's coding for proteins. Number 2, explain what Hox genes are. Hox genes are a group of genes that control differentiation of cells in zygotes. Number 3, recall the types of mutations that lead to frame shift mutations. The loss or gain of a nucleotide in a sequence leads to frame shift mutations. Remember that chart, that table in the notes. Number 4, compare missense mutations and nonsense mutations. Both mutations are the result of a substitution. In missense mutations, the DNA code is altered so that it codes for the wrong amino acid. In nonsense mutations, the codon is changed to a stop codon, and translation terminates early. And lastly, number 5. Demonstrate how the DNA code for a protein can be mutated by inserting a nucleotide at one point in the sequence TTT, CAG, GAC. Now, of course, didn't really specify which or where. However, an example here, if we look, there's an added G. Right there it is, where my crosshairs are. Now, this is an insertion. So what's going to happen is the amino acid, so this is the DNA sequence. This DNA will then be transcribed to messenger RNA. Well, if this occurs, this mutation, this DNA strand will transcribe for a different mRNA strand than this one would. Now, obviously, the first codon here would be the same. However, anything after that will be affected because now we have GCA instead of CAC, and so on and so on and so on. So it would affect everything moving forward. And that's it for 12.4 Active Reading Review. This is Mr. Case signing off.